My honor to introduce Dr. Janine Bijsterbosch. She's professor of radiology at Washington University in St. Louis. She earned her Ph.D. at the University of Sheffield in the UK and her postdoctoral training at Oxford University. She currently directs the Personomics Lab at Washington University within the Computational Imaging Research Center in Radiology. She's a pioneer, as you'll hear, in leveraging large neuroimaging data sets to identify personalized signatures of brain organization that can predict critical mental health outcomes. Her innovative work applies computational approaches and integrates imaging with multiple sources of clinical and other data. I have had the great pleasure of collaborating with her on our new R01 project called Harmony, and we're combining 4 connectome data sets for depression with other large imaging data sets. And I'm thrilled that she could join us today. So please welcome her to the stage, and she will present on computational neuroimaging of population mental health. Welcome.
Good morning, everyone. Thank you for that kind introduction and for the invitation to be here today. I'm looking forward to the rest of the day. It's a really exciting schedule. And I'm excited about the opportunity to tell you a little bit about the work that we do in the lab at WashU. So as we've talked about already, and the first talk really set the stage for this, scientific data sets are growing, and this image is showing that in my own work. Each dot here is a publication as a function of year of publication on the x-axis, and then sample size on the y-axis. And I had to put that in log scale to show the growth from 20 subjects early on to tens of thousands of subjects later on. I was just telling our plenary speaker, people used to be impressed when I told them that, but now our data sets are actually not as big as some other data sets. And this is a table from one of our recent review papers, kind of summarizing different types of data, many of them big data in one way or another, ranging all the way from sort of deep phenotyping in a small number of participants, really repeated measures, to epidemiological data that is trying to be population representative. And most of the work that I'm going to talk about today is going to be at that right-hand side of the scale. And as Dr. Williams mentioned, one data set and one project that I'm really excited about, but won't really have time to delve into more today, is the Harmony data set. This is an NIH-funded project where my lab serves as the data center for this. And we're taking 4 pre-existing cohorts in the space of mental health, each of them containing about 150 to 250 patients, and harmonizing them and combining them into one superset. So our lab uses multiple different data sets, but the data set that we've used the most so far, and that I'm going to be talking about mostly today, is the UK Biobank cohort. And data are still being acquired for this data set, but the goal in the end is to have neuroimaging data in 100,000 individuals. We currently at WashU have data for about 45,000 people sitting on our servers. And this is what's called a prospective epidemiological data set, which means that there's no specific disease focus. This is not a mental health data set in the way that Harmony, for example, is. But there's also no clinical exclusion criteria. So this is also not a healthy cohort. And participants are followed up until death unless they opt out of the study, and patient health records are being shared, as well as lots and lots of other data available on these participants. And so I think this is really exciting in the context of studying mental health. I'm reminded of the study that I recruited for when I was a postdoc, a traditional study of funding for 100 people, recruiting and noticing that a lot of participants who had real mental health challenges were actually not eligible for the study that I was doing for various exclusion reasons. And so I think these population data sets offer a lot of opportunities to study the transdiagnostics, the comorbidities, the subclinical individuals, and many other aspects of mental health. There are also important disadvantages, and I want to spend a few minutes, first of all, talking about those and how we think about trying to address and overcome those. And so one of the disadvantages is a lack of clinical diagnostics. The UK Biobank does not include a structured clinical interview for diagnosis. There are some things we can do. They do have ICD-10 disease codes derived from clinical health data, so that's one thing we can use. And other people have developed other categories of binary DSM-style classifications based on self-reported data. There's also too many healthy controls. Like I said, this is not a mental health specific cohort. So the majority of people have minimum scores or neuroticism or other measures relevant to depression and mental health. And so in many of our studies, we do some sort of screening to try and obtain a more clinically relevant cohort out of the UK Biobank. This is a work in progress. We're still very much thinking about the best way to do this. This example study, which was one of the first projects we did in the UK Biobank, we used this one question, screening question of whether people had ever gone to their doctor for mental health as a screener. There's also a lack of validated instruments. I like to tell people that the UK Biobank measures everything you can possibly want to look at. I have collaborators who are interested in hearing, and I can give them variables that are relevant to hearing. The catch is that they probably don't measure it in the way that you'd want them to measure it. And that's because they're trying to do a lot of different things. And so the UK Biobank includes the patient health questionnaire, which is a pretty standardized and well-known measure of depression severity, but it's not available in all participants. And it was done at a different time point from when people came in for the scanning. That's usually years apart for most participants from when they're scanned. So we wanted to use what we call the Recent Depressive Symptom Scale, or the RDS, which is a measure of 4 questions that were asked to participants on the day of scanning. And this is, of course, not a standardized measure. And so we went out and got some new data outside of the UK Biobank, new cohort of participants who completed this measure, as well as some more well-known measures of depression, twice 1 week apart, so that we could look at the similarity as well as the retest reliability to validate this. So those are just a few extra hoops that someone has to jump through to kind of use the measures that are available in the UK Biobank and make the most of them. Okay, so now that and I'm sure you can come up with more disadvantages of the UK Biobank. I'd love to hear them. Let's talk more about it. But for the rest of my time today, I want to really dig into the advantages of this type of big data set. And I want to talk a little bit about how we think in the lab about using and leveraging those advantages of population mental health data. And I think when you've got 45,000 people, we've got tens of thousands of summary measures or features derived from neuroimaging data. We've got thousands of non-imaging measures, many of which are clinically relevant. The temptation is to use what I call the kitchen sink approach, which is basically put it all into one big analysis and see what comes out. And the thing is, you'll find something, and I bet it will be significant because we've got all this power. It will probably even be reproducible. But whether it's interpretable is a different question. And so we try to move away from this approach and try to think of ways that we can use the UK Biobank in a more research-driven, sort of hypothesis-driven approach. And so we've come up with 2 different philosophical ways of thinking about this type of data that projects in the lab tend to fall into. The first one is what I'm calling the needle in a haystack approach. And this is where we're using the UK Biobank, treating it kind of like a database from which we can pull interesting and unique participants. And so projects that fall into this approach tend to only have sample sizes of a few hundred participants. And I'll give you some examples of studies like that in the rest of my talk. The second approach that we take is what I'm calling the selective packing approach. So when you're going on a trip, you technically have access to anything in your wardrobe, anything in your house. But what you put in your suitcase depends on where you're going. And when you're going to a safari in South Africa, you pack different things from when you're going on a city trip to Paris. And so we take a similar approach for these types of studies. We might use most or all of the participants that we have available, But the variables that we pick and the computational methodology that we select is really very much driven by the research question, the hypothesis, the end stage of the trip. And so I'm really excited about this idea of analytical study design. I think there's a lot that you can do when you take a question and come up with the data and the methods later. And that's really the approach that we take in the lab. And I think that's, even though it's a data-driven, it's a computational approach, it can be hypothesis-driven. And so we really focus on trying to do studies that offer interpretability while still having that rigor in terms of validation and replication. So now that I've laid the groundwork, I want to spend the rest of my time in the talk today kind of very briefly introducing 3 different studies that we're just wrapping up in the lab right now that fall into these different categories to hopefully illustrate to you a little bit how we're thinking about this type of data. And so the first study is about heterogeneity in depression. There's been a lot of amazing papers looking at subtypes of depression, and this study is not one of them. The goal here was not to see if we could find subtypes. The goal rather was to try and understand the kind of complexity of the sources of heterogeneity that we see within depression and kind of better understand how those different ways in which participants differ from each other might interact with one another. So this is work that was led by my graduate student, Kayla Hannon. She's amazing. She's actually graduating this spring. So if you need a postdoc, reach out to me or to her, and I'll be happy to introduce you. She realized there's many different ways in which patients can differ from each other. One way is in terms of the clinical presentation. These are 2 imaginary patients with the same diagnosis of major depressive disorder. And as you look through their symptoms, you might see they have one pretty vague symptom in common. These are not real patients, but this is a realistic example given the diagnostic criteria for depression. But beyond this type of clinical heterogeneity, there's also another example. You might have 2 individual patients with the same diagnosis and also the exact same clinical presentation, but they might still differ in terms of the neurobiological pathways or mechanisms that give rise to that clinical presentation. And so Kayla was interested in differentiating between these interactions between clinical and biological heterogeneity. And that leads to different theories of how that might happen. So you might have one theory, the one-to-one mapping idea, where you have subtypes that differ from each other, both in terms of their clinical presentation and in terms of their neurobiological pathways. And so in this case, whether you look at the clinical data or the neuroimaging data, you would expect to find the same answer, the same subtypes. There's another theory where there might be a many-to-one mapping, where some of the same clinical presentation might result from different types of neurobiological pathways. And note that these 2 theories are not mutually exclusive. You can have one-to-one mapping in some subtypes and many-to-one mapping in other subtypes. And you could even come up with more complex kind of interactions. So we wanted to test this hypothesis, and we took this approach of really looking at clinically dissociable groups of individuals. So we looked at 3 different symptoms of depression: low mood, anhedonia, and somatic symptomatology. Most individuals with depression present with a mixture of those symptoms. They're highly collinear in the overall population. But when you've got such a big data set to begin with, you can find those interesting and unique people who are high in one symptom and happen to be low in the other symptoms. And you can play the same game with other clinical kind of characteristics of depression, such as chronicity and acute severity. Again, those are usually highly collinear, but you can tease them apart when you've got this big data set to begin with. And at the bottom, we have a heterogeneous group, and this is our typical depressed cohort, which has a mixture of all of these things. And so we performed normative modeling to look at deviations from the norm for different structural and functional neuroimaging measures. And I'm going to show you the results in a minute. We looked at which measures differ between these 2 groups, between these clinically homogeneous and heterogeneous groups. And these were our predictions. We thought that these dissociated, these tightly controlled clinical groups would have larger magnitudes of deviation than the heterogeneous group, thinking that heterogeneity is what's contributing to what we found in the literature with small effect sizes and inconsistencies between papers. And we also thought that the homogeneous groups, or the dissociated groups, would have different normative patterns. So these are the results. You can stare at these for a long time, and we sure did. But I'll draw your attention to a couple of results here. The last bar in each of the column, that dark red one, that's our heterogeneous comparison group. And so you can see that that bar is smaller than most or even all of the other bars in many of these imaging features, confirming our hypothesis that you get larger effect sizes when you disentangle clinical sources of heterogeneity. So that's kind of getting at this one-to-one mapping idea. We also see that there's very different patterns between the groups, so even some of them going in opposite directions, which is suggesting that there's a lot of diversity in terms of the neurobiological pathways contributing to different aspects of one disorder. We went a little bit further to try and test this idea of many-to-one mapping. And so Kayla performed subtyping or clustering within each of these clinically homogeneous groups. This figure is showing the stability of these subtypes. And you can see in some of these groups, you find really stable subgroups or clusters. And honing in a little bit more on one of them, we found within the acute impairment group, 2 different clusters. And when we looked a little bit more, it showed that these clusters differ in interesting ways from each other. So one cluster, the red cluster, has preserved neurobiology, so relatively high still fractional anisotropy, gray matter volume, cortical thickness, and performs cognitively similar to healthy controls. But we also have a second cluster that has reduced fractional anisotropy, gray matter volume, and cortical thickness, and lower cognitive score. So we think this might be really interesting in relation to the idea of depression being a risk factor or potentially even a prodrome for cognitive decline in older age. So I've shown you that we found in this example larger effect sizes in homogeneous groups and different patterns of brain variation for different symptoms. And so that shows you some evidence for both one-to-one and many-to-one aspects of heterogeneity. And hopefully it shows you an example of like how we can use such a large database to draw interesting groups of participants that we could never dream of recruiting if this was a study we were doing from scratch. I want to give one other example in this idea of the needle in a haystack approach. And this is work by a postdoc in the lab, Wei Zhang. She's actually on the faculty job market. So again, if you have any opportunities, let me know or let her know directly. And so she was interested in the differentiation between state and trait depression. So how are your symptoms now versus what is your general predisposition to being depressed? And these things are usually highly correlated. Unfortunately, past depression is the best predictor of future depression. However, if we're thinking about biomarkers, trait and state biomarkers would actually be used in very different ways. You know, a trait biomarker could be used before the onset to estimate risk and work on prevention, whereas a state biomarker is essential if you want to measure change, if you want to look at tracking over time or efficacy of a certain treatment. And so we played the same game of looking for those rare individuals where we could break this collinearity and dissociate state and trait depression. So you can find individuals who had high depression at baseline at recruitment, but happened to report low depression scores several years later when they were scanned. And again, you drop in sample size, but you still, if you start with so many people, you still have a decent sample size to work on. And we played a similar game to get a group of state depression dissociated from trait depression. These are some of the results. So these are trait-specific dissociated from state markers of depression. This is network amplitude. So this is a functional MRI marker. We did not find any evidence for state correlates dissociated from trait of depression, and we can talk more about why that might be. We were a little surprised to find the visual cortex here, the visual network. That's not what you find often in the literature. And we're actually currently doing some follow-up work to try and understand that result further and see if it replicates in some other big data sets. Okay, so with the minutes that I have remaining, I want to show you one last study that we're doing that falls into this other category, the selective packing category. And this is moving a little bit away from depression, looking more at transdiagnostic work. So this is work that's led jointly by graduate students Ty Easley and Lexi Luo in the lab. I'm keeping them for a while. The goal of this study was to try and classify multiple different diagnoses. And so using neuroimaging features, try and predict patients and controls. And we wanted to do this to look at the disease specificity. You know, there's a lot of machine learning papers out there that predict patients versus controls, but they usually just focus on one diagnosis, or maybe they do bipolar and schizophrenia, but it's rare to see a broader analysis of more diagnoses. And so we had that opportunity in the UK Biobank to do this more broadly transdiagnostically. And we also wanted to do some benchmarking. As I mentioned, in the UK Biobank, participants are going to be followed up until death. And so this type of like going back to their neuroimaging data and predicting what diseases they ended up developing 5-10 years down the line is going to be work that's probably happening more and more. And so we wanted to do some benchmark and lay the groundwork for that. These are the 17 different diagnoses that we use for this work. These are from the ICD-10, from chapters F and G, which are brain-relevant disorders. And so I'm going to show you the results. These are 17 different machine learning models predicting in each case patients versus control. We did also do a multi-class prediction. I don't have time to go into that. And in each of these 17 different models, we have the patients and an identical number sample set of completely matched healthy controls. We're data rich, so we can match them identically on age, sex, and head motion, for example. And so chance is at 0.5. These are the results. So in color are the best predicting feature set. In gray is all of the feature sets. We tried a few different feature sets. And so you can see here that the category of demyelinating diseases was significantly predicted above chance based on structural imaging features, and depression was significantly predicted above chance based on functional, but in this case, actually not structural features, which is an interesting result. Overall, these prediction accuracies were a little lower than I had expected. And so we think that that might be related to some reliability issues of the ICD-10, both in general and also some of them particular to the UK Biobank. And I'm happy to touch on that more if we get a chance to. And so with that, hopefully I've been able to convince you that we can do interesting things with population data in the context of mental health. Really what our lab is trying to do is not do a poor man's job at research that could be better done in a dedicated study. You know, there's when you have full control over the recruitment and the variables, then there are some studies that are better suited for that type of scenario, but we're trying to do studies that you couldn't do if you had to do the recruitment yourself. And hopefully you've seen some examples of that here. I'm really excited about this idea of analytical study design and these approaches. So with that, it just leaves me to thank our funding, our collaborators, and thank the amazing lab members that I get to work with every day. Thank you.
We have time for 1 or 2 questions. The microphones are to the side, or just put your hand up if you need a roving mic. If there are none, we have plenty of time in our panel discussion session at 2 p.m. to go into detail. There is one. Go ahead.
These neuroimaging studies based on some neurochemical metabolic factors specific to the brain cells, or
there's different types of measures that we get from the neuroimaging. So you can do different sequences to measure different things in neuroimaging. Much of what we've done so far is about the size of regions. So the gray matter volume, for example, within a region, or the function of regions. So how much two different regions interact with each other, or how strong a network is. We are also now starting to do some work in inflammation. So the degree of inflammation in the brain and in specific brain regions, which I think is really exciting. We're also starting to do some work on GABA and other neurotransmitters. So there's a lot of different measures, but most of what I presented today was structure and function measures. Thank you.
So notice there's a big difference, say, on EEG with different age groups. And it seems like that factor can override lots of other factors. So have you noticed that? And are you sort of accommodating for that?
Yes, we have. There is certainly a relationship between the depression severity score and age in the UK Biobank. And I think it actually goes down. I think as people get older, their depression scores get lower, if I recall correctly. We do control for it. So when you do the normative modeling in the first study that we did, that's kind of setting, that's as a function of age. So that's normative distance from people of the same age. And in the last study, we did age match controls or age and sex match controls. So yes, we are cognizant of that and controlling for that. Yeah.
So with the scanning, you're not just the depression scores.
Yes, right. With the scanning. Yes, absolutely. Thank you.
Okay. Thank you. Thank you for such an inspiring presentation. We look forward to more discussion in the panel session.